Hello everyone, my name is Harold Gomez, and as Dr. Alexis already mentioned, I'm a senior student at St. Thomas University. I was fortunate to work at Dr. Plunke's lab during the summertime, and I was involved in the expression of the chondroitin sulfate proteoglycan Neurocan in the adult zebrafish. So, I want to start off by giving you some background information on what chondroitin sulfate proteoglycans are. I know you have already heard something about them. This is a family of molecules which are characterized by having a protein core, we can see here in, okay, as you can see here in red and in yellow. They also have these glycosaminoglycan chains, which are nothing else than sugar chains that are involved in the interactions of these molecules with different proteins and other molecules. So they are known as modulators between the interaction of the cell and the extracellular matrix. What they do is that they bind to specific protein receptors found in the plasma membrane of the cell, for example, neurons, and they do so by using their glycosaminoglycan chains. By doing so, they modulate different activities. One of them is known to be as inhibitors of the expression of the growth of the axons in the neurons. We can see here a neuron that is being repelled by a CSPE spot that we can see in red, and how it goes by without really getting into it. We know that CSPEs are expressed in human, in mice, in rats, and in zebrafish. So they are expressed along a very wide range of organisms. Previous work in Dr. Plunke's lab was able to show the expression of the CSPEs at the injury site after several days of injury. We used an antibody called CS56, and we can see these bright areas are the CSPEs in the spinal cord and in the brain. However, I need to emphasize that this antibody does not recognize a specific CSPE. It's only specific for the family as a whole. However, we still know that in the zebrafish, for example, there is a Neurocan, brevican, tenascin as some of the members of this family that are expressed. I'm going to focus more on Neurocan since it's the center of my research. Why Neurocan? Well, Neurocan is a secreted CSPE. It's found primarily in the extracellular matrix surrounding the cells. It's also primarily present in the central nervous system. What is this? Why is this important? Okay, most CSPEs are found primarily in the articulations, in the joints. They serve as a cushion for all the motions that you do. However, Neurocan is found in the central nervous system, which might infer that it's more closely linked to regeneration of axon growth. We also know that Neurocan is highly conserved between mice, rats, and human, and it has, as all the other CSPEs, a protein core and these glycosaminoglycan chains. The glycosaminoglycan chains are unique for Neurocan as they are unique for the other members of the CSPE family. So at this point, we see ourselves, well, I saw myself with two questions. The first one being, is Neurocan expressed in the zebrafish before an injury happens? Or is the expression of Neurocan a result of an injury? So my hypothesis was that Neurocan is actually upregulated after an injury in the central nervous system of the adult zebrafish. So in order to test my hypothesis, I start off by dissecting the tissue from the zebrafish, then performing RNA isolation to extract the RNA from the cells, then do a cDNA synthesis from this RNA, and then performing RT-PCR to amplify the signal, and finally doing gel electrophoresis to analyze the expression of the different DNA fragments. So the tissue that we harvested was from uninjured and injured zebrafish. We did the RNA isolation by using a minikit called the Qiagen RNA Easy Plus minikit. This kit has a series of buffers that are allowed to separate the RNA from the other proteins and molecules that are found inside the cells. However, RNA, as many of you might know, is a very unstable molecule. It can degrade really easily. So what we do in order to make it more stable is we convert it back into cDNA. This process is just the opposite of transcription, in which DNA is converted into RNA. To do so, we need to do this reaction, we need a series of reagents. One of them being a special enzyme called reverse transcriptase. This enzyme is the one that allows the transcription to take place backwards. We also need a primer that's going to start the synthesis of the cDNA sequence. And this primer, we use oligo-DT. We also need free nucleotides that can be attached to the chain to build a complementary DNA sequence. So the reaction starts off by the oligo-DT binding to the poly-A tail of the mature RNA. After that, this allows for the reverse transcriptase to bind to the 3' region of the oligo-DT primer and start adding the free primers in a complementary way to the mRNA that we're using as our reference. However, once we are done synthesizing the cDNA, the amount of cDNA that we have from the mRNA is not really much. It's not really a workable amount. So what we do to make it more workable is we amplify specific regions of DNA in which we're interested. We call this reaction a polymerase change reaction. And it's a standard protocol that is used in many labs, but in this case we were interested in amplifying only the Neurocan gene, since we are studying Neurocan. So PCR consists of 3 main phases. During the main phases, DNA is exposed to a high temperature. These high temperatures allow for the DNA to unwind and open its structure for the primers and the nucleotides to bind. Once the DNA structure has been opened and has been denatured, the first primers bind to both extremes of the gene that we want to synthesize. These primers are the ones that are going to set the beginning and the end of the gene that we want to amplify. And finally, the DNA replication steps takes place in which an enzyme starts adding the nucleotides in a sequential way, and we do the same cycle for 35 times. This allows for the DNA to grow in an exponential way. So, and then we go into analyzing this DNA fragment to see its expression by running it through an agarose matrix, doing a technique that is called gel electrophoresis. These techniques, what it does is it uses the negative charge found in the DNA to separate it by size. The size we measure using the number of base pairs, and we use a DNA ladder as a way to reference the size, the size of the band that we're looking at. The DNA ladder that we use is a 100 base pairs DNA ladder, so each band corresponds to a 100 base pairs increment. So we go from 100 to 200 and so on. So what results did we got after doing all of these experiments? So we found that Neurocan is present in the CNS of the zebrafish even before injury. We found that in spinal cord, doing a series of different concentrations of the Neurocan gene, we found, of the cDNA, we found an expression of the Neurocan at the level that we were expecting to find it, between 400 base pairs and 500 base pairs. We have, in lane number 6, we have a negative control in which we then use the reverse transcriptase that was supposed to synthesize the DNA from the RNA. What this does is that allows you to see if there's any contamination coming from genomic DNA. And we can see there is none, and that these ones actually correspond to the Neurocan. We also found the same expression of Neurocan in the brain, and we can see that there is no contamination from another kind of DNA or other molecule. So previous work done in the lab by France showed that Neurocan is also expressed in the brain of the zebrafish after the injury. We can see here that after 7 days, we can see some level of expression, and after 14 days, we can see a clear band for Neurocan. We use beta-actin in lane 1 and 2 as a positive control. What does this mean? We know that the Neurocan is expressed in beta-actin, so we use it in a way to see if the band that we're getting is in the right place, and also, in case we don't get the band, to see if the primers and the all reagents and all the reactions took place in the right way. So, all this information that we got was contrasting with what we know from mammals. Mammals have shown to be to show upregulation of the Neurocan after an injury. However, we need to notice that the RT-PCR is a qualitative test. It's not quantitative. It can only show expression. It doesn't show the amount or level of expression. In order to do that analysis, we need to use another kind of technique that is called real-time PCR. So for future directions, we want to confirm this result by doing more of these tests. We also want to test whether the Neurocan protein is expressed and not just the mRNA before and after the injury in the central nervous system. I would like to acknowledge Dr. Plonke for giving me this opportunity, Dr. Alexis Tabanes Castillo for always being there supporting me, the U.S. Department of Defense, the Minority Science and Engineering Program, France for having so much patience with me, Megan and Gabriel, two of my close lab partners that work with me in this project, and the rest of my lab peers. These are my researchers.
Any questions?
Yes.
Could you go back to your second result slide of your gels?
Sure.
You may have said it, I might have missed it. What is the significance of the beta-actin being expressed less at 14 days and the Neurocan being expressed more at 14 days?
Well, it's not this is not a quantitative test. So the brightness of the band doesn't necessarily correspond to expression of the Neurocan in the in vivo in the animal, okay. Right? We only use beta-actin as a positive control, meaning that we only use it as a way to know if the reaction took place in the right way, and if that, if we see a band, if that's the right band that we're supposed to see.
Okay.
Yes.
It seems like what could account then for the difference in intensity?
Well, it might be, okay, what accounts for that difference in intensity is the amount of DNA that is loaded into the gel. It might be that, that's all I can refer to as that.
And in your PCR slide,
sure. PCR. Here.
Right. Could you show us which are the, your molecule, which are the 5' end and the 3'?
The 5' end, okay. I don't really have a label in here.
In other words, how does PCR really work?
PCR is really, the step of replication is just as the step in replication in DNA. The primate binds to the different ends. One binds to one end and the other one to the other end of the, and it replicates in both directions. So one replication takes place in one of the chains of the DNA and the other one in the other chain of DNA. One since always. Since one chain is complementary to the other, you're going to have the same DNA fragment at the end. So replication takes in the 5 to 3 prime direction. Does that answer your question, or?
What happens to the original molecule?
The original DNA molecule? They are involved in the same, they're included in the same sample. They don't get degraded or anything else. They're just incorporated as part of the DNA that we're replicating. It shows that the new DNA sequences that we're making have one old part of the DNA that we're replicating and a new one that's being just replicated. Is that better?
With regard to the RNA,
yes.
How did you handle the RNA originally to maintain it pure?
How do we make it pure? Yes, we use this kit that has a serial buffer. It has RT, different buffers that actually separate the RNA based on the pH and different interactions with other molecules. And it has a column that serves as a filter for the RNA.
So it's known that even sweat has RNAs which will degrade.
No, it doesn't. If we had, no, it doesn't have. It's actually we work in an RNA-free environment.
Right, that's what I was going to get at. So what is that environment? Have you
We, how do we get to that? Okay, we have an RNA-free solution that actually degrades RNAs. So we work with that solution in this environment. And when we process these RNA that we extract, we try to just minimize the exposure to any kind of degradative interaction. Yes.
How did you check for RNA integrity?
How do we check for RNA integrity? Well, there are two ways we can do it. The first way, we can measure RNA concentration by using a spectrophotometer. Right? Or we can, if we don't see an expression of the cDNA, that also can be a signal that the RNA was degraded.
Did you run a gel for the RNA?
For the oops, not really. We don't have a gel for RNA because if we use it, it's just degraded. We wouldn't stand the temperature that is necessary for that. Just exposing it to a current will degrade it since it increases the temperature. Any other question? Thank you so much.